He currently works as he currently works as a senior assistant professor at Arjuna University in Ethiopia and specialized in toxicology, biosafety, and risk assessment for chemicals, herbal medicine, as well as nanomaterials. He has conducted a toxicity study of fish, lab animals, as well as human samples, and skilled in computational tools for drug design and toxicity. Dr. Kumar sir holds a PhD in nanobiotechnology as well as toxicology and PG diploma in toxicological patterns. He has published in numerous research papers and is a member of several professional organizations, including Society of Toxicology and the International Association of Forensic Toxicology. His expertise include molecular analysis, big data, and alternate methods for animal testing. Welcome, sir. I welcome you to our faculty development program. We are excited to have here. Once the Benjamin Franklin said that an investment in knowledge pays the best interest. We are committed to providing you with a valuable learning experience. Thank you for joining us. I want to extend my welcome to Dr. Suresh Kumar sir for this session. Welcome, sir.
Thank you, sir. Thank you, Prem Kumar sir, for this for the introduction about me, and thank you so much. Many thanks to the organizers also, and thanks to Dr. Amal sir for this invitation and organizing this event.
Professor Suresh Kumar sir, sorry to interrupt you. Before handing over the session to you, I would like to introduce our participants to you. Here we have participants from different fields, from biotechnology, biomedical engineering, chemical engineering, as well as from computer science background. Also, we have a composition of different universities and colleges all over the India. So this is for your information, sir. And now session is yours. Please, sir, please introduce us. Thank you.
Thank you, sir. Thanks for letting me know about the audience, and so happy to be part of this Pan India workshop on FDP. So I'm so happy for that. Let me share my screen, and please tell if you can able to see my screen.
Yes, sir.
So can you see my screen, please?
Yes, sir. It is
Yes, sir,
perfectly
you
viewed. Yeah, you can see your screen, and your audio is very fine, sir.
Okay, sir. Thank you. So good evening to one and all present here. So myself, I am Suresh. So I am working in Ethiopia in Abamanch University for past 7 years. So my expertise is on general toxicology as well as forensic toxicology. I do some of the research in environment as well. So I'm a board certified toxicologist, and today I would like to give a topic which is something relevant to life sciences and biomedical science people. Even I have included some of the topics that will be relevant to people from computer science also, because in recent times the role of artificial intelligence is getting more, and there are a lot of scope for people from computer science to explore the various avenues in biotechnology also. So I hope so that some part of my slides would also benefit the people from non-life science background as well. So today my topic will be on insights into conventional and contemporary methods in toxicology testing. So to just start my presentation, before starting my presentation, these are the contents I would like to discuss today. So some of you might be knowing about what is toxicology. Some of you might not be knowing it. So it's good to tell what is toxicology, what is the need to do a toxicity study, and how do we do a toxicity study that comes under toxicological risk assessment, and what are the methods available to do toxicity. And every time we go with animals for testing the toxicity of any chemical, drug, or any medical device or something. So why don't we try with something else? Of course, there is no much alternatives available for the animal-based testing models, but now in recent times we have some new alternatives have come, and it has been put into practice for regular pharmaceutical. It has been put into practice in some of the CROs, contract research organization, who do this sort of studies. Even US is also promoting, US, Europe governments are promoting this kind of alternatives for animal studies. So I'll cover some part of my presentation will cover that. And the last one will be the in silico approaches, whatever is available and what is commonly used by people also. I would like to discuss during my presentation. So to start with, to people who knows what is toxicology, it's well and good. But people who don't know, it is a study of adverse effect of chemicals or any agents on the living organism. So adverse effect means usually we tell it's harmful effects. You know, the basic understanding of toxicology is generally based on the dose, the concentration that only determines a poison. The simple example, when we take anything in excess, it is poison. That's what we even in our Hindu mythology, or even in our traditional way of medicine also, we believe that anything in excess is always a poison. So the toxicology centers around this concept. And Paracelsus is the first person to describe this kind of the term dose makes a poison, and he is also considered as a father of toxicology. So in toxicology, we'll be studying about what is the adverse effect of any chemical or biological agent or anything on any organism. Mostly it should be on living organism. So it can be a microorganism, or it can be any biological organism. It can be human being or animals as well. So in toxicology, as I told before, anything in excess is poison. So it's always, we have to understand that when we have everything in balance, when we have everything in equilibrium, it is good. When something goes above that, always we have to face the consequences, as like what we have seeing in this picture. Too much of loading is not good for the animal or for the system also. The same applies for our system. So what will happen if too much is coming into our body? Of course, the body will react to the too much thing that is coming into our body by causing some adverse reactions. So who are these toxicologists? So I'm telling I'm a toxicologist. Someone will say someone will be working on biochemistry or biomedical science, and he claims I'm a toxicologist. Someone will be working some nano pesticide detection based methods. Someone will be working on that. Someone will be working on some small algae in the environment, and he'll try to find out what happens when a chemical is coming into the environment, how it affects the small organisms in the environment, trees. So many people claim they are toxicologists actually. So why? Because toxicology is not a single field. It involves a lot of information from diverse fields. We need diverse expertise to do a toxicological risk assessment. So these are the peoples we need in case of toxicology. Mostly biochemists will be part of it. Chemists will be there who knows good about the chemical structure and how the chemical structure will cause the adverse effect. So chemistry is one of the very important component of toxicology. Pathologists, suppose we give a chemical to animal and we wanted to see what happens to the liver, what happens to the heart, what happens to the blood. So we need a trained pathologist who has a good about how it affects the cells. So these things will be done, these assessment will be done by a pathologist. Then we need people who are working on oncology, cancer researchers, because we know some of the chemicals are known to cause cancer. And it is also very important that whatever the drug or chemical we use it for medical purpose or for therapeutic use, it shouldn't cause cancer. That is a baseline understanding of, that is a very, very basic important thing we should think in mind. We should think when we develop a drug or when we give a drug for some treatment purpose. So the drug, if you are giving a drug for treating pain and if the drug is able to cause some cancer, then there's no point in giving the drug for treating a pain, right? So cancer research is also part of it. Veterinarians, because medicine is not only with human being, it is also with animals also. We have veterinary medicine group of people who are specialized in veterinary medicine. And it is also a part of toxicology, where studying the adverse effect on animals. We need medical doctors if you're going to work with human subjects. We need cell and molecular biologists who are able to study the basic, the very up to the cellular level. If you want to see what the chemical is doing to a cell or organ or something, we need a cell and molecular biologist to study that. We also need engineers, because engineers are very important for devising the instruments, developing experimental setup. For this case, engineers are important. Mathematicians for mathematicians and statisticians for doing the statistical analysis of the data that generates out of toxicological study. Lab technicians. Of course, we also need animal care providers, because most of the toxicity studies are done on animals, like rats, mice, and so we need animal care providers. Most of them are veterinarians, but sometimes they don't have to be a veterinarian. Even a 12th grade passed person who has a good 12th grade passed person who has an interest on animal welfare, or who has an interest in taking care of animal, can also be a part of the toxicological study. In most of the CROs, we have people like that who are working for taking care of animals. So these are all the people who are very important for doing a toxicological study. And these people, the input that comes out of from all these people is very important for finding out the toxicity of a chemical or whatever it is. So what these toxicologists will do, we try to assess and understand how the chemical is going to affect our body. How the chemical in the sense, it may be a drug, it may be any chemical, or it may be heavy metal, whatever it is. So how it affects our body. To do that, we need an expert person. The expert persons are the one, these are all the people who are experts, and these are all the people who participate in this study, and they try to understand the following things. First, we have to understand the mechanistic role of the chemical, how the chemical affects the body, where it goes and bind, how it changes the level of some enzymes, or level of some proteins or biochemical biomolecules in our body. So to do that, we should have a complete mechanistic understanding, which is very important to know the toxicity of a chemical. Then some toxicologist will make sure that the chemical or the drug, whatever we are using, is safe for us. It will not cause any adverse effect. Mostly it will be in pharmaceutical industries. These people are very important. And also in food industries, food additives. The food additives that we add for the food should be safe for us. It should not cause any adverse effect. Recently, I think INS3, new food additive, red color dye, has been banned in some part of the world, claiming that this dye can cause erythrocin we call. It can cause, it is probable to cause cancer, colon cancer, in people who consume this dye. It was identified through some of the rodent studies, but it's not 100% accurate. But still, FDA has banned a dye called INS. INS3 is Indian nomenclature. The dye name is erythrocin. So like that, the toxicologists will try to ensure that the chemicals, whatever we add in our day-to-day use, is safe for us. And also the drugs, whatever the drugs we use for some treatment or for therapeutic purpose, should be safe. And suppose if a chemical enters into your body and the chemical is causing some adverse effect, some harmful effect, so what should we do for that? How do we have to treat the disease? How do we have to treat the symptoms that comes because of the adverse effect? So all these things will be determined by clinical toxicologist. And also one of the prime importance of toxicologist is also the public health. So when it comes to the public health, it's very important that they get safe food and safe water, because that is a very integral part of everyone's life. So the toxicologists, like environmental toxicologists, they ensure that the food and water we take is safe. The level of the chemicals or the level of some heavy metal should be always in the minimum threshold, required threshold level. If it exceeds, then it's not good for safe drinking for human or animal use. The other applications are forensic, which is mostly related to medical legal types. So you know that when after a person die, if there is any suspicious of any poisoning, then the samples will be taken from the dead person, and a technician will do the analysis whether the chemical is, whether the poison or any drug has been taken, and which is a reason, which leads to a death of a person. So these things will be identified by forensic toxicologists along with forensic doctors. And also we do have environmental applications of some of the toxicologists also will participate in many of the environmental applications, like oil spills, assessing the risk of oil spill in the environment. So these will be done by this kind of environmental toxicology specialized in environmental application. So here in the right side, I have given some of the fields in toxicology: forensic, general. General is a general thing where anyone can take part of it. So genetic toxicology, regulatory toxicology. So these are the people who, regulatory toxicologists are the people who are in FDA, Food and Drug Authority, or even in our India, we have regulatory authorities in our Environmental Protection Agency. So what they do is they will try to collect information from all the toxicologists, and they try to form a regulation guidelines. So this is a acceptable level for a particular chemical to be present in water, or this is acceptable level for a chemical to be present in air or in soil. So these will be done by regulatory toxicologists. And medical and clinical toxicologists are mostly concerned about welfare of humans. Mainly human-related toxicology will be handled by medical and clinical toxicologists. So these are all the work done by the toxicologist. And as I told you, toxicologist is not a single discipline. Multiple people are involved in it for this science. So now let's come to the toxicological risk assessment. So I've been telling continuously about adverse effect, causing some adverse effect, something will cause some bad thing. I'm telling about the same thing again and again. So how can we quantify, how can we measure the risk? Risk is nothing but whether the thing is going to happen or not. The probability that the particular thing is going to happen or not. It's a kind of probability, risk. So we have to find out in a quantifiable way that this chemical will cause this effect, or this much amount of chemical will cause this much effect. So that whole thing will come under a category called toxicological risk assessment, TRAs. So the risk assessment can be done for drugs, risk assessment can be done for chemicals, general chemical, the chemicals that we use in day-to-day life. Risk assessment can be done for devices that we use for medical applications. So there are diverse disciplines in TRAs. So today, as most of my title is related to drugs, so I will be focusing only on the risk assessment related to drugs. So when it comes to toxicological risk assessment, we have three important things. One is risk assessment, which is called hazard identification. So you need to identify the hazard, whether it is hazard or not hazard, what hazard it is going to cause, what adverse effect it is going to cause. So that is called hazard identification. So we have different types to do the hazard identification. Whether you can directly give the chemical to a animal like rat, and you can find out whether the chemical is causing harmful effect or not. We can do it. Or suppose we don't have animal, then we can go for some in vitro methods. In vitro means lab based methods, lab grown method. So for example, the cells, we can able to grow the cells in the small plates. So instead of giving to the animal, we can give it to the cells, and we can identify whether the chemical is going to cause toxicity or not, which comes under in vitro methods. So you can do this thing. Or I don't have a facility to do on cells, or I don't have a facility to do on animals. That is the advantage of recently we have machine learning methods have come. Recently we have a lot of AI tools, artificial intelligence tools have come, where we don't have to go for the lab work. Just by in silico based, by computer based method, computer assessment, using computational tools, we can identify the toxicity of a chemical, we can identify the hazard of a chemical. It's all probability based or statistically based, we can do it. And we also go for epidemiological studies, where we test it in a group of suppose we collect the information from a population, and we'll find out how the chemical has caused the problem to the population. That comes under epidemiological study. So all these things will come under risk assessment. So once we identify the risk, okay, this is what the risk is going to happen, we have to identify the dose response relationship. Dose response is nothing but it's simple. We give 5 or 6 concentrations of drug. Example, suppose with the animal I'm studying, I give 5 or 6 concentration of the drug, and I will try to find out the response of it. So in case of toxicity, we study the adverse effect. If in case of drugs or something, if you want to see that benefit therapeutic effect of a drug, we just go for, suppose we go for the particular pharmacological effect. For example, I'm giving paracetamol, and I'm giving different concentrations of paracetamol, and I'm trying to find out which concentration is causing the therapeutic effect, which concentration is causing the adverse effect. As you know that paracetamol, if we take 6 capsules of paracetamol per day, which is approximately 650 or 1 gram of paracetamol, if we take 6 capsules a day, it's going to be toxic to the liver. So we establish a kind of relationship in a form of curve or in a form of a graph that this much amount of chemical, if you take, it is going to cause adverse effect, toxic effect. This much amount of chemical is going to cause beneficial effect. That type of relationship, that type of curve or that type of study is called DR study, DR curve, dose response relationship study. So which we usually identify initially. Then we'll go for exposure assessment. Exposure is nothing but in which way the chemical enters into the body. So we should know what are the different ways a chemical can enter into the body, a drug can enter into the body. So in that case, we have to identify what exposure route. The route will be mostly oral, because most of the drugs we take through oral route. Sometimes we take it through inhalation. Sometimes the drug can also enter through skin. Sometimes it will enter through direct. Some drugs we have to give directly to the body, like route direct route of entry is also there. So all these things should be assessed. Earlier in dose response relationship, we will try to find out what is the effect the drug causes in the body. That is dose response relationship we are going to identify with respect to the dose and effect. But here we are going to identify in different routes of exposure, whether through mouth, what is a DR relationship, if through skin, what is a DR relationship, that we will try to find out by this way. So that is called exposure assessment. So once we have all this data, then we go for risk characterization. So we have to identify what is the risk based on the values data we have. We have to identify what is the characterization of the risk, whether it is highly toxic, medium toxic, or less toxic, or what is the dose required to be given to the population to reduce the toxicity. So these are all comes under risk characterization. So together the whole framework is called toxicological risk assessment framework. So as I think we have seen this in our day-to-day life in some of the chemical bottles. Suppose we use some solvents for our laboratory purpose. We can see in the backside of the bottle, we'll see this kind of labels. So they are called GSH labels, the globally harmonized system of classification of toxicity. So what the scientists have done is they have studied the chemical and they identified that this chemical is going to cause this kind of effect. It may be a general physical effect or it can be a biological effect, but generally these warning symbols will give an idea about what is the effect of the chemical, either on the body or on a physically we can find. For example, if you have a danger skull symbol, it means it's going to cause toxicity, acute. It's going to cause harmful effect. So suppose if some drugs, if we see a drug with star, suppose we see a symbol with a star-shaped symbol in the chest region of a person, then it indicates that this is going to cause different types of toxicity in a person's body. We call systemic toxicity. Systemic means it is going to affect the entire body system, right? So these are all the things we have to be very careful whenever we see the label of any chemical. Most of the time we don't see it, but it's good to practice to do that. The second, if you can see that in the back of your medicines, they will write if you take more than there will be some uh, small what you call paper will be there kept inside the drug telling statutory the warning will be there where they mentioned what are all the adverse effect the drug can cause, right? So these are all some of the things which comes from the toxicity studies. Only by doing the toxicity risk only they're able to identify these are all the things that can cause to a person. So we are going to see a little deep about toxicity studies, where it is useful. So generally for drug approval process, suppose if you want to, you identified a chemical, you identified some chemical which is causing some good biological effect. For example, the chemical is treating the body pain. Simple. When we take the chemical, it is going to treat a body pain. We are identified by some simple research. We extracted from bacteria or we extracted from a plant, and we know that this chemical is good for reducing the body pain, right? we call it as analgesics. Okay, so it's a good analgesic effect, we'll tell. So how do we go with the drug approval process? Some of you might be knowing it, but maybe for people who are not from biological background, it's good to explain this. So this is how traditionally a drug goes through developmental process. The first will be the screening, where, as I told you, we extract the chemical from a plant or we synthesize the drug by ourselves using chemical methods, and we have the drug in hand. It's not a drug yet, but we have some chemical which we have some beneficial effect. So what we'll do, once we have the chemical identified, the structure identified, the physical properties, everything identified, then we go for non-clinical or preclinical studies. Preclinical means before going to the treatment with the patients or before going to the population, human population, we have to study in the animals. And also we can also, before going to the animals, we do it with cell lines. So first in vitro tests are very important. We do the in vitro study, we find out what is the toxicity, then we go for in vivo, which is called animal studies. We go with the animal, we'll find out the toxicity, and we collect all this information. Then with all this information and the structural and chemical information about the drug, we go for IND approval, investigational new drug application. So we have to submit with all these things, we submit to FDA or the regulatory body, whoever it is. So what the regulatory body will do, the regulatory body will assess all this information and they will give approval. If the drug is okay, if they find that the drug is going to cause some good effect in a population, or they find it is having a great potential, then they permit for going for clinical studies. So in clinical studies, we try to test it in a population, phase 1, phase 2, phase 3, where the number of population will increase depending on the phases. So once the clinical studies are done, then we go for new drug application with the clinical data in the population. Clinical means whether the drug is going to reduce the body pain when we give it to a patient or someone. Then what is the data we collect from them? How much time it is effective? How long it is effective? What is the cost of it? What is the adverse effect we see in the population? So everything we collect from this study, clinical developmental study, and we collect all this information. And then we collect the preclinical and clinical data. Together with that, with the clinical data, we go for new drug application. So once we submit the new drug application, the group of scientists or regulatory toxicologists will deeply analyze this one, and finally they give approval. So once approval, we have to go for post-market surveillance, where after the drug goes to the market, we have to do the surveillance of whether the drug is causing any adverse effect, or whether the product is really genuine, whether the product is really working. If there is any issues that are happening in the market, what are the litigations we have to manage. So all these things will come under drug approval process and post-approval process. So here, the toxicological studies are very important in preclinical phase. The preclinical phase only we have toxicity studies done most of the times. So the same way, this is not in the scope of the presentation today, but just give you an idea. Suppose we have a medical device. Suppose we identified some nanomaterial, a scaffold. Scaffold we have identified by some method we identified as scaffold. The scaffold is used for wound healing purpose. Suppose if we have a cut in our skin, we put the scaffold or a gel. It is going to cause the wound is going to be cured immediately. So we identified something like that. Is it can we directly use it for application? No, not at all. We have to go through biosafety evaluation. So for that, we have different types of studies are there. Analytical testing is there. We have to identify what chemical is present in that scaffold. If it's a nanomaterial, what metal is present, we have to identify. Then we have to go for biocompatibility studies. We have to keep the scaffold in animal, and we have to see whether how the animal reacts to the scaffold, whether any allergic reactions are there. So that we'll study in biocompatibility study. We also study the toxicology of the medical device. If the medical device is having some plastic chemical and that chemical is causing some toxicity to the liver, in that case, the device cannot be used for treatment purpose. It cannot be used for medical treatment. So this is also a different aspect of toxicity risk assessment, where we study the safety of biological devices, which are intended for some treatment purpose, human treatment or animal treatment. So here comes a toxicity testing scheme. So generally, how do we do a toxicity study? So here the left side, mostly we use animal for toxicity study. That is a conventional way we are following it. You can see almost 10% of the animals are used for toxicity research. 34% of the animals are used for biomedical research, different types of biomedical research. And of course, in R&D of veterinary drugs, suppose you develop a drug for veterinary treatment, most of the animals, 44.4% of the animal is used for that kind of veterinary related research. So what is very clear is animals are used for research. There is no alternative for animal in research. As that's what the conventional testing will say. So how to go for toxicity testing in animals? Suppose we want to go for conventional way. So the left side I have given a flow chart where we identify a chemical, we identify the test material. We have to find out what is a chemical, what is the active chemical present in it, what is the structure of it, what is the physical property. Everything should be clear. Then we go for literature analysis. Directly we cannot inject a chemical into animal, or directly we cannot go and test your drug in the animal. So before that we have to go for literature search, we have to go for structure activity assessment. So that is called QSAR. There is a tool called as QSAR or chemoinformatic based methods. I will explain it later. So what we try to do is, if you know a chemical structure, by different methods if we're able to find the chemical structure, it is very easy to predict the toxicity of a chemical, because if there are similar chemicals like what we have in hand, it is very easy to identify the toxicity of a chemical. That is called as QSAR method. QSAR, quantitative structure activity based relationship method, or some chemoinformatic methods are also there, which are helpful to do this kind of studies. So once we know about some information about the drug, then we can go for acute toxicity. Acute means in short-term toxicity study. We give for 3 days or 1 day or 4 days, and we'll find out the toxicity of a chemical. So during this process, we'll identify multiple parameters. So we have to identify the pharmacokinetic, we have to identify the pharmacokinetics or toxicokinetics. We have to identify the pharmacodynamics or toxicodynamics. So what is pharmacokinetic dynamic? A very simple way. Pharmacokinetics, what we define is what the drug will do to the body. Suppose you take a drug, pharmacokinetics tells that what a body will do to the drug. Suppose you take a drug, how the drug is getting absorbed, how the drug is entering into your blood, how the drug goes to the target organ, everything comes under pharmacokinetics or toxicokinetics. So once the drug has entered into the target, for example, it entered into a cell, liver cell, what the drug will do to the cell or to the body, that is called pharmacodynamics, or if it is an adverse effect, then we call it as toxicodynamics. So all these things we'll try to find out in toxicity studies. So along with the acute toxicity studies, we also identify the other type of toxicity like reproductive toxicity, whether the chemical is going to cause adverse effect on the reproductive organ, whether it is going to cause adverse effect on the development of a child. Suppose if a woman is pregnant and we give a drug to that pregnant woman, whether the drug is going to cause damage or any adverse effect to the developing fetus, all these things will come under developmental and reproductive DART study or teratogeneity study. And also we also try to assess whether the chemical is going to cause, the drug is going to cause cancer in the body. So a battery of tests will be done along with the acute toxicity test to find out the toxicity of a chemical. So these are all the different tests as I was telling you. And I think I can go. So acute toxicity is short term. Subchronic means less than 90 days, 3 months. Chronic means more than 90 days. So we give the chemical for more than 90 days and we'll find out what the chemical is doing to the body. So that is called chronic toxicity testing. So there are many testings, many studies are there. I don't want to go into detail. So I'll just give a brief idea about these are all the different tests will be done. So we have genotoxicity assessment. Genotoxicity means whether the chemical is going to affect the DNA. If the chemical is going to affect the DNA, then there are more chances that it may cause mutation, and that mutation may lead to some problem in the particular protein, or it may also lead to cancer. So all these things we will study by different tests. So we have Ames test, we have mouse lymphoma test, we have transgenic rodent mutation test. So these are all the different tests we do conventionally by recommended guidelines. We do all these tests and we'll confirm that yes, this chemical is going to cause genotoxicity, or this chemical is not going to cause genotoxicity. So to do that, we have to do at least 2 or 3 tests compulsory. So we also need to go for cancer studies. So a minimum cancer study, suppose you want to identify the carcinogenicity potential, whether your chemical is going to cause cancer, a minimum 2 year study is required, minimum 1 year, but some guidelines, then the guideline people will go up to 2 years also. So till 2 years you have to wait to know whether your chemical is going to cause cancer in the animal. So we confirm by different method. We take the organ and we'll sacrifice the organ and we'll see whether the cancer growth is there. We sacrifice the organ and we'll take the organ and see whether the cancer growth is reduced because of treating a drug. So everything will be done by using the carcinogenicity study. So minimum 2 years is required for this kind of study. So in mutagenicity, we do this kind of thing. Then we do skin irritation testing. Previously there was a rule that if you are shipping a chemical from a foreign country to some other place, you have to be sure that the chemical is not a corrosive or not a irritant. Irritant means if the chemical falls on your skin, it is not going to irritate your skin, not going to cause some damage to your skin. It does not give any allergic reaction to the skin. So if to identify that, previously they were using animals for testing it. So they take an animal, they apply a small amount of chemical on the skin, and they will see whether the chemical is going to cause irritation. So this was done before, but nowadays the thing is banned and we have a replacement for that. Then we use beagle dogs for doing the respiratory toxicity study. So whether the chemical enters through a nose, suppose you wanted to take an inhaler drug, how the drug is going to enter into the body, what is going to do. We usually test on beagle dogs. An ocular irritation, suppose if the chemical falls on your eye, what will happen? So we usually use chicken eyes are recently used. We can also use with rabbit eyes. Mostly rabbit eyes are used for this kind of study. So you can see from all these things, animals are going to suffer a lot during these studies. And we also have developmental reproductive study, we call DART study, where we try to identify in each stage of reproduction how the chemical is going to affect the reproductive ability, from the development of gametes or to the development of embryo, or the fertilization or the development of embryo or the development of fetus, or after the child is born, until during the weaning stage. So in every stage we try to simulate this, we try to do it in animal, rat, and we'll try to find out how the chemical is going to affect these stages. So it's a very big study. I'm not going to go into deep about it, just give you a overview of how the scientists are trying to find out the toxicity of a chemical by different means. So far from whatever I discussed before, what is very clear is chemicals, most of the studies are done on animals, right? So recently, 10 years before, 15 years before, due to the lot of suffering that happens in animal, team has come up with a thing called 3R concept. So 3Rs are very important now and recently. If you are going to do any experiment on animal, the guidelines strictly ask you to follow the 3R principle: replacement, reduction, refinement. Replacement is try to use another method instead of using animal. So that's what the replacement says. Reduction, try to use less number of animal, because in many of the studies they usually say we have to go for fixed number of animal to do the experiment, to do the toxicity study. So the guidelines nowadays are changing. They're trying to tell us, they set a guideline that try to use less number of animal or statistically relevant number of animal for the study. And refinement is a welfare. So whenever you're doing a toxicity study, see that the animal is not going to suffer much. The animal is going to suffer, try to give them better treatment options so that they feel less amount of pain. So this thing we call it as 3R principle, which is very important for toxicity research. So this is the international perspective, how the governments are trying to remove the animals from the research. Different things have happened. Even in Netherlands, by 2025, they plan to phase out all the use of animals in drug testing. In cosmetic products, whatever you're using, without going through animal testing, a cosmetic product cannot come into the market, because cosmetics are the one we are going to use on the skin. If the cosmetic is going to cause skin irritation, the people are not going to buy that. So most of the cosmetic companies, they test on the animal, they find out that it's not causing any toxicity, allergy or something, then they give it to the market, then they sell it to the market. So recently in European countries and all, a label has come. The companies have taken a different step where they do not use animals for testing the cosmetics. They use with some other alternatives for that. like in vivo methods or some in silico method or in vitro method, people are using it to do the kind of toxicity testing. Even India has tried to push the alternatives for animals in biomedical research. In 2019, some news have come related to that. Still, development is going on in this. So is it really possible to replace animal in the research? It's a long-term process. It's not going to happen immediately, but in recent times, it's happening. How? Let us see. So spineless solution for 3 hours. So can we test on animals that don't have spines, invertebrates? Possible. How far it is relevant? Let us see. So, this is a widely used toxicity model organism called zebrafish. It's a small fish where it has the genes present in the zebrafish is more similar to human being, not 100% similar, but some similarity is there. So because of that, all the human disease models has been mimicked in zebrafish. So instead of trying on human being, instead of trying on some animal, why don't we do it in zebrafish, try to find out the toxicity, and we get an idea, then we can go for further testing. So by this way, we can reduce the number of animal we are using it. We can try to replace the animal from vertebrate to invertebrate. Even then, this is still in developmental stage. Regulatory bodies have not yet fully given approval for this. Scientists are trying to develop more models using zebrafish to be used in future. So what is the advantage of this is, in case of developmental studies, the zebrafish embryos are very easy to monitor in microscope. The development can be the entire 24-hour development we can easily monitor in a microscope. So that's the reason why zebrafish has gained a lot of attention. You give your chemical to the embryo and see how it affects the development of an organism. Widely used, still people are trying to use this for doing a developmental testing. Then brine shrimp, of course, brine shrimp is, I just put this slide because for people who don't have access to zebrafish, who don't have access to anything, they can try with brine shrimp for studies. But please consider that if you want to study the environmental toxicity of it, you can use brine shrimp. Mainly if your chemical is going to enter into the sea or somewhere, marine water, then testing it on marine Artemia salina, brine shrimp is a good option. So as I told you, brine shrimp is very easy to do. The studies are there is a very regulated studies are available. There are OECD guideline is available. We can follow that. And it's very easy to do in any laboratory setup. People who don't have access to that kind of big laboratories, they can follow this brine shrimp assay, but it determines only the environmental. It is mostly useful for environmental toxicity, not for human related work. This is from my thesis, whatever I done my PhD. So how the brine shrimp is, I had a nano chemical, I developed a nano pesticide, and I used the brine shrimp to identify the toxicity of it. So just it gives idea how the chemical is affecting the development, how the chemical is affecting the adult shrimp. We can able to identify from this microscopic studies. The next one is the Daphnia. Daphnia is a microcrustacean. It is widely available in green waters. Suppose you have a stagnant water, you can see Daphnia in that. So people have collected Daphnia, and it is a very good bioindicator for environmental health. Suppose you have a water system and you wanted to know whether the water is good or not, just take the water and see how many good Daphnias are there. If the Daphnias are there, which means the water is good. Because the Daphnia is a clear indicator of the toxicity to the water. Many ecosystems, for many of the water aquatic ecosystems, freshwater ecosystems, Daphnia is a very good bioindicator. It's very easy to do in lab. There are some kits available. Even there are some laboratories in India, South India. They are giving Daphnia culture, so you can use it. You just need a small plate to culture the organism, and you can do the testing of a toxicity testing of a chemical. It can be done by any person, provided you have a little training in it. This is just a study to give you an example how Daphnia can be used to stimulate the food chain in the environment. Just for reference, I've given it. Then another organism is C. elegans, which is also a small crustacean, microcrustacean, small worm. It is actually nematode. It is widely studied. Even a person has got Nobel Prize for studying something in this thing, because it can be used for development of many neurodegenerative models can be developed using this organism. This is also one of the promising organism for toxicity testing in environment. and this is a larva called Gallerilla melanola. So this can be used for antimicrobial testing. So like you inject the drug into your into the into the uh, abdomen of the larvae, and you see the signs that is happening. So there are some indexes are there. There are some indexes to calculate how the drug is effective on our body. So you inject the microorganism, the pathogen, and you can inject the drug, and you can see how the drug is affecting on the larva. This is also one of the thing, but it's not widely followed. But still, it's a good alternative organism for baseline studies. And this is a very ideal thing for genotoxicity study. Though in regulatory, you don't have much role, but if you want to do it in your lab, we can try this. It's very easy to do. With the tip of the onion root tip, we can take it, we can stain it using some dye, and we can, because the chromosomes in the active tip or growing tip of onion is easily visible. So anyone can do this thing if you want to do a toxicity testing of any chemical that you've developed in your laboratory. It's widely used by many people. So this is how all the mitotic stages can be seen clearly in onion root tip. So we can use this for our study. You can use in your lab. Then Drosophila is also one of the widely used model for developmental studies. It needs a kind of practice. It's a fruit fly, but still it is also one of the alternative. Silkworm is another alternative. We don't have any ethical concern. It's cost effective, but we cannot do it for a lot of drugs, but some drugs can be tested using this kind of thing. These are all the overview of what are the in vivo infection models available. Whatever I discussed, the same thing I given in an overview. Now, how these invertebrates are used in toxicity testing? Of course, it takes time because an invertebrate may not exactly resemble the human system, but in a preclinical developmental stage, the invertebrate testing can try to reduce the number of days you take it for testing on animals. So if you have a prior information in doing an invertebrate, that give you idea whether the chemical is going to work on rat or not. So by that way, we can reduce the number of days we use it for research, number of cost, number of money we put in this kind of research can be reduced. So still there is a lot of hope for this invertebrate and toxicity testing. Although, what is the future of it? Although invertebrates are not absolute replacement to animal research, still they reduce the number of live vertebrates we use in our research. So still there is an increasing demand in this kind of spineless animal testing. Now, these are all the things I told about using the non-invertebrate model. What about the real human relevant model? So we have a lot of new test guidelines have created where artificial skin has been created. Artificial, suppose the epidermis, the skin is having 3 layers, epidermis, dermis, hypodermis. The epidermis itself is having 5 layers. So scientists are able to grow these 5 layers of skin in a lab. So using a specific test guideline for 497, we can apply that chemical on the lab grown epidermal skin and we can identify the response. So this is widely followed. Even regulatory authorities are accepting this kind of studies. Phototoxicity studies where we try to find out whether the chemical under the influence of light is going to cause it's becoming a toxic chemical or not. That we can identify by guideline 498. So like that, there are new guidelines have come in recent times which are good alternatives for testing on animals. So this is the example I told you about skin irritation test. We have 3D tissue animal models where we don't have to go for animal. Just by growing the skin cells in the tissue culture plate, we can able to do the testing. It's commercially available. Of course, it's expensive, but it's a good replacement thing. Now, the last part of my presentation will be on in silico approaches, which I think it will be useful for many people. So in a world way, conventional way, when we go, we don't have any, suppose we identified a chemical from a plant. We don't know what the chemical is going to cause to the body, but simply we just know that this chemical is going to do something. So what we will do, we'll take the chemical, we will give it to the animal and we'll see it, right? So most of the time, animal systems are dynamic. Sometimes it will give a false negative, sometimes it may give false positive. There are a lot of chances for error, right? So a lot of animals are needed for this study. And if you want to do a lot of battery of tests, for example, acute toxicity, mutagenicity and other thing, imagine how much animal is required, how much time is required, how much cost is required. So as an alternative, we have in recent times in silico approaches. in silico combined with in vitro approaches. So in silico is computational tools, right? So what we do is computational tools, using computational tools, we try to predict the toxicity of chemical, whether it is probably toxic or what level it is toxic. So based on that in silico information, we get from information we collect from in silico analysis, it is very easy to go for animal testing and reduce the number of animals as well as reduce the time required for preclinical development. So that is a new conventional way of toxicity testing. So it will try to reduce the number of days required for preclinical studies. So in that we usually follow a system toxicology approach where we have multiple systems, like we try to find out the genomics, where the effect on the genes, we try to find out the effects on the proteins, we try to find out the effects on the metabolites, and everything we put it together, we try to model it in a one system. And when we input our chemical, we can be able to find out the toxicity of the thing easily in an in silico way. Sorry, just 5 more minutes, I'll finish it. So system toxicology.
There was an interruption.
Sorry, sir. Yeah, system toxicology approach is recently, it's getting a lot of attention because, as I told you, you don't have to go for animal directly. Just you input. The main thing what you have to do is you should know the chemical structure. Structure is a very important thing to be noted. If you do that, the rest of the things can be done by computational system analysis itself. 50% of the work is already done by this thing. So that's what we call system toxicology approach. And the in silico approaches, for example, we can input our chemical to a database, okay, and in the database we have already known toxicity for list of chemical. So we have our own chemical. So what the computational tool will do is will try to match the structure relationship, your chemical structure and the already available chemicals which are known to cause mutagenicity. So it will try to match from the database and it will try to give a score. Yes, this part of your chemical can more prone to cause toxicity. Your chemical can cause 50% toxicity, genotoxicity. Like that, it will try to give you some idea using an in silico approach. So these are all the examples. We have OECD toolbox. It's freely available. Only thing, just need some practice. So you give your structure, it will try to screen your structure with lot of chemicals which are already deposited in the database, and it will try to give you the results, whether it's going to cause genotoxicity, or whether it's going to cause environmental toxicity, what will be the acute toxic value. Everything we can find out in a theoretical way. So that's one of the recent advantages of using computational tools. So anyone can try OECD toolbox. The training is very easy. You can just follow the YouTube, or if you go to the OECD toolbox website, they will try to give you all the information how to do it. It's self-explanatory, self-learning. Then we also have another thing called Swiss ADME, where we input the chemical structure, or we give the SMILES format. SMILE is one of the representation for chemical. So that you can identify SMILES format, you can easily get it for a PubChem or some database. You input the SMILE structure. This online tool will try to give you how much percentage of toxicity it will cause, whether it will cause blood-brain barrier, whether it will cause genotoxicity. A lot of things it will predict by computational tools. Just we have to input the chemical structure, that's all. The same way, ML. So machine learning has become, in recent times, it's becoming more popular. So what we do is we take two types of data. One will be your training set and one will be your prediction set. So what we do is we go to the PubChem. PubChem is a site where we get information about all the chemicals. So we go to the PubChem, we retrieve information about some chemicals. Like for example, environmental endocrine disrupting chemical that is going to disrupt on the hormones. So we get all information about what are the known chemicals, we take it. We give it to we create algorithm, we give it to it. At the same time, we are using some different types of algorithms. I'm not very good in ML based tools, just I know the basics of it. So using ML based algorithms, we're using a training set, we'll try to find out the toxicity of the unknown chemical which we have with us, right? So this is done by ML based approaches. PubChem is one of the database where we can retrieve information about the chemicals. VEGA is a tool which is software is used for ML related database construction. ML machine learning construction can be done using VEGA, which is a freely available software. And the last one is adverse outcome pathway. So what is adverse outcome pathway is? This is a simple way of representing an adverse effect that is happening on a system. So previously, suppose if you give a chemical to a rat, the adverse effect I will identify by knowing whether the rat is dying, or whether the rat dies, or whether there is an infection, or whether there is an allergy in the rat. Visually, I have to see everything, or I have to collect some blood and I have to measure the biochemical changes. So what the scientists have done is, many people have done already. So with available existing information, they made it into a simple way of representing the molecular adverse outcomes, right? So what simply they do is, suppose, for example, I'm telling that a chemical is going to cause cancer. So if the chemical is going to cause cancer, we have three adverse outcomes, right? If the cancer is there, what will happen? Inflammation may be there, metastasis may be there, the immune cells, the cytokine levels may be increase or decrease. So suppose we know the adverse outcome one, suppose it's going to cause inflammation. So what I'll do is, I'll do a specific bioassay in the cell lines. Suppose it's going to cause cancer in the liver cell line. So I took Hg2 cell line, right? I give my chemical and I'll do the particular bioassay to prove that this chemical is going to cause this adverse effect. So instead of going directly with the animal, by knowing the adverse pathways, so we have different adverse pathway for different types. For inflammation, we have adverse pathway. For metastasis, we have a different adverse pathway. For genotoxicity, we have adverse pathway, right? So we measure the key events in that adverse pathway by using some bioassays, so that without testing on animal, just simply by doing this in vitro based studies, we can easily identify the toxicity of a chemical. AOPs are getting a lot of attention. We call adverse outcome pathways. So for this, it's very important to collect a lot of information, put it together, and we have to develop an assay system, and we have to prove the concept. That's what we call AOPs. This is example I've given for adverse outcome pathway for nanomaterial. Suppose you have developed a nanomaterial, you wanted to study the effect of adverse effect of nanomaterial. Just go for the guidelines AOP 173, which gives all idea how we have to do the testing in the cells. Rather than going on animal, we can just test directly in this. Right. So at last, so what I'm trying to tell is, recent times computational tools have gained a lot of attention. But it's very important that even though you work on computational tools, it's very important to test on any biological system. So biological system, we can go for in vitro studies. In vitros are little compatible, easy to do. Compared to, of course, in vivo is easy to do because doing in rats are easy, provided you have ethical approval, facility, everything. But if you don't have it, better we can outsource it to a lab that is having in vitro facility. Computational tools can be done by the lab of your lab, and you can give it to the other lab and you can get the result done. By this way, the research, whatever you're doing, will be very productive, and it can also easily go to the go to the human, it can go to the population easily. Of course, we cannot directly reach the population, but the data, whatever you do in a systematic way, by this way, it will help to helpful for a lot of for future research. Yeah. So thank you all for the patient listening. I hope so I've given a brief introduction about toxicology and the different testing method, as well as how computational tools are used. So thank you so much for this opportunity. Thank you for the organizers as well as the team. And I'm ready for questions. So if you have any questions.
Thank you, Professor. Yeah, thank you so much. Your presentation was very nice, and we are highly enriched with the toxicology studies details. And now the session is open for the discussion. Participants, if you have any questions, please you may unmute yourself and ask, or you may get into the chat.
So can I stop sharing, sir? Shall I
Yes, sir,
stop
please.
sharing?
Yes, sir. Yes, sir. We can. You can stop sharing. Participants, if you have any question, yeah.
Thank you, sir, for your informative talk, sir. I want to know how to analyze whether the dye toxicity is reduced after treatment with some nanoparticles or something like that. How to analyze whether there is reduced toxicity after treatment or not?
Okay, ma'am. I can understand the type of study you're doing it. So you have some chemical dye, and you have a nanoparticle. So you're trying to find out the nanocomposite or nanoparticle is able to absorb the dye, or it is reducing the toxicity of dye, right? You wanted to know how to identify that. It's better way you can try with the onion method, ma'am. Onion phytotoxicity study, because phytotoxicity study is easy to do. Like I told you, there is onion root tip assay. So what you do is you grow onion in your lab. When the roots are started to grow, cut the tip of the root. Sorry, by the time you have to keep four beakers. In that four beaker, you add your different concentration of dye and the different on the nanoparticle what you are going to treat. You just add it, and after some time, what you do is you know that okay, in this tube, in this beaker, the dye would have been reduced. In this beaker, 50% it have been reduced by the nanoparticle. Like that, you have some idea before, right? So what you do is in the same beaker with different concentrations of dye or different concentrations of nanoparticle added, you try to grow the onion root in that. And once the onion root started to grow, you take a tip of the onion root, you put it in a slide, apply some dye. We have common dyes are there. Um, Even Zimsa stain is enough. So you can add a Zimsa stain, and you can just observe under microscope how much cells are having positive mitotic cells. Ma'am, are you from biology background or?
Yes, sir. Yes, sir.
Yeah, so I hope so you can understand what I'm trying to say. So you just go through onion genotoxicity study.
Okay, sir.
I think that will be the easy one to work. That's what I suggest, because you are working on dye and you're working on nanoparticle. Anyway, these two things are not going to be for medical purpose or something. It's going to be some environmental purpose, I believe. In that case, it's better to go for onion root tip assay, where the publication also will be easily accepted and it will have good value. And you are also trying to find out the genotoxicity potential. So the toxicity potential also you can identify by this way. There are a lot of papers related to this also.
Okay, sir. Thanks.
Yes, ma'am.
There is a question in the chat box, sir.
Chat box.
At the bottom right. Yeah.
Yes, sir. I think, thank you, Dr. Brinda. Dr. Priscilla has asked a question. Is it mandatory to know the algorithm? It is mandatory to know we are from biotech background. What are the possibility to learn such things? Okay. See, actually, this is usually we face this kind of issue because we are not more exposed to the computational based method. Recently, we are in the era of ChatGPT, right? So in ChatGPT, we can easily find out the codes for whatever the thing we are going to do, machine learning based method. So what we can simply do is there is a software called Vega, WEGA, right? You can just Google it, how to do the machine learning, chemoinformatic machine learning analysis in Vega. There is a protocol is available. So just you follow the protocol, the algorithm, everything is already inbuilt in the system. So just you have to you have to give your training set and you have to give your unknown set, and the algorithm, everything will automatically, whatever you choose, it will work and it will give you the result. So to know best about the different types of algorithm, I think the basic information is enough to do this kind of studies. There are different types of algorithms out there. Suddenly I'm not able to pick up because a long time back I did this. But it's easy to do. It's not mandatory to know the each and everything. But in this new era of chat GPT, I think it's easily we can able to crack the algorithm very well. And we have GitHub, GITHUB, GitHub is there, where a lot of people have deposited the algorithms, whatever they tried in the GitHub. So we just have to go there, they're given explanation, follow that, and you can get the results. So that's how I do. So it won't be a big issue, I hope so. But if you still have a doubt, you can just reach me by email and I can help you with some tools, or there are some trainings available I can recommend for you. Thank you. I hope so I answered your question. And you don't have to worry about learning ML. Anyone can do it. It's not very difficult. Thank you, Dr. Ravishankar. Thank you for the wonderful thing. Thank you, Priyanka ma'am.
Thank you, sir. Thank you so much. We are about to end the session now.
Thank you.
I take this opportunity to thank Professor Suresh Kumar sir for his valuable time and knowledge sharing. It is really helpful for us as participants, as mentioned here. We are really appreciate the knowledge what you have shared, sir. And thank you so much for this. And with your permission, if we could share the PPT, we can share with the participants. Sir.
Sure. I'll do it. I'll do that, and feel free to reach me for any clarification related to toxicity studies. I would welcome. I have I given my email ID and I'll give my phone number. I have WhatsApp number.
Yeah.
So if anyone is having any doubts in toxicity studies, they can feel free to contact me, because I am from biotech background. I'm not from pharmacology side. So from biotech background, I become a toxicologist. I got certified, and I'm practicing it in recent times. So I can understand how it's difficult for biotech people to follow this, or even for people who are non-biotech also. It's very easy to follow toxicity studies, provided with some little experience. So I feel free to contact me for any help. Thank you all. Thank
Thank you so
you,
much, sir.
Amar.
Yes, sir. Yes, sir.
I'll
I will
share it
share
to you.
definitely. I'll share your details to the participants.
Yes, sir.
Right, sir. Thank you.
Thank you, sir. Thank you.
Participants,
So,
participants,
shall I
we are
leave?
about to. Yes, sir. Please, sir. Thank you. Thank you so much. Yeah.
Thank you.
Participants, we are about to end the session now, and the next session will be starting immediately, because we will wait. We'll have 5 minutes time. By sharply 7:30, we'll be starting our 7-8 session. Welcome, Professor sir. We have our resource person, Professor Ranganathan sir, with us. Sir, just now our session is
I will
getting
share my
over.
slides, sir. 1 minute, I will share my slides, sir.
No problem, sir. Can we have a 5 minutes time for a little break? Just now the session is getting over. 7:30, can we start, sir?
Yes, definitely, sir. Before that,
Yeah.
shall I share my slide?
Definitely, sir. We can check it. Yeah, please,
yes,
sir.
sir. Yes, sir. Yes, sir.
Participant, you may take a 5 minutes break. We'll start our session by 7:30 sharply. Yeah. Dr. Prabhu, sir. Dr. Prabhu, sir.
Yes, sir, I'm there.
Yeah, please, sir. Perfect, sir. Your slides are visible.
Yes, sir. Okay, sir.
Very good.
Let me to change my slide, sir. 1 minute,
Sure,
sir.
sir.
Can you see the second slide, sir? Objective?
Yeah, it
Yes,
is
sir.
okay, sir.
Okay.
It is
sir.
okay.
Present fuel system? Okay, sir. Thank you, sir.
Participant session 7 feedback link available in chat box. So all of you complete the feedback form. Good evening to one and all present here. So next, the session 8 will going to start. So I am happy to read the profile of the session guest, Dr. S. Ranganathan sir, Professor, Department of Biotechnology, Anna University, Chennai. Sir completed in UG BTech Chemical Engineering in Annamalai University and MTech in Annamalai University Chemical Engineering, and the PhD was completed in Anna University AC Tech Campus, Anna University, Chennai. So sir having a 25 years experience in teaching and industry experience, and sir having received many honors and awards from the various institute and agency. Sir received through T.M. Narayana Pillai Chemical Engineering Prize from the Governor of Tamil Nadu at 1995 and received Best NSS Program Officer Award from Anna University, Chennai, and received Best NSS Unit Award from Anna University, Chennai, and Bright Student Award from Government of Tamil Nadu at 1986. Sir received Young Scientist Award from DST Science and Engineering Research Board, New Delhi. Sir also received the Best Faculty Award from Science and Technology Award 15. Sir also received Active Researcher Award from Center for Technology and Development Transfer, Anna University, Chennai at 2015. Sir also received Outstanding Scientist Award from Center for Advanced Research and Design. Sir also received a Lifetime Achievement Award from International Research Award on New Science Invention from Government of India at 2021, and Sir also received Best Researcher Award for International Research Award on New Science Invention by Government of India at 2022. Sir also having a 53 lakhs amount of sponsored research, and also he was completed successfully at last 10 years. And Sir also having 8 PhD scholars, and he was also completely PhD scholar. 18 PhD scholars also completed, and ME MTech students 93 project guided, and ME MTech project also going 8 number of project going. Sir, 3 books was published and 18 book chapter also published, and 193 national international reputed journal also published. So SARS also having a very good knowledge biochemical research like biodiesel production and bioproduct and biomass and biofuels. So once again, I welcome Dr. S. Ranganathan. Sir, sir,
Yes.
This is your session.
Yes, sir. Thank you, sir. Can you hear my voice, sir?
Yes,
Can
sir.
you hear
Yes,
my
sir.
voice? Yes, definitely, sir.
Yes, sir.
please.
So before start the session, I want to introduce about participant. The participant from the various institution, various universities around the India, from various division like chemical engineering, biotechnology, pharmaceutical technology, and as well as computer science and mechanical department faculties also joined in this ATAL sponsored FTP, sir. So thank you very much.
Thank you, sir, for giving me this opportunity and also introducing me over here. Shall I start my lecture, sir? Hello, sir?
Yes, sir. It's clear.
Yes, sir. Thank you, sir.
Yes, sir.
Sir,
Your voice
yes,
is
sir.
audible.
Yes, sir. Thank you, sir. I'll start my lecture, sir. So, respected coordinator, Dr. Maria Amalraj, sir, Associate Professor, Department of Biotechnology, State Institute of Technology. Beloved co-coordinator, Dr. N. Prabhu, sir, Associate Professor, Department of Biotechnology, State Institute of Technology. Distinguished faculty members and research scholars, and my dear participants from various universities and also institutes. Very good evening. With immense pleasure, I would like to express my sincere thanks for giving me this opportunity to present my lecture on role of computational tools in optimizing the production of biodiesel from marine macroalgal biomass in the AICTE ATAL sponsored faculty development program. Before my presentation, I would like to give a small introduction about my lecture. Due to the depletion of fossil-based fuels, research on renewable and environmental friendly fuels such as biodiesel has received a lot of importance in recent years. The biggest benefit of the biodiesel is its environmental friendliness over gasoline and petroleum diesel. Biodiesel is usually produced from oily crops such as mustard, soybean, sunflower, and palm. Moreover, the availability of the oil crops as the source for biodiesel production are limited. Therefore, it is necessary to find a new feedstock suitable for biodiesel production, which does not rely on the edible vegetable oil supply. One alternative to the conventional oil crops is the algae, because they contain oil suitable for esterification transesterification reaction for the biodiesel production. Biodiesel production from algae is widely considered as one of the most efficient methods. It appears to represent the recent renewable source of oil that could meet the global demand for transport fuels. The other side, the occurrence of algal blooms greatly disturbs the ecosystems. For the first time, biodiesel production from marine macroalgae was carried out in our lab, and our group has succeeded in producing biodiesel from marine macroalgae such as Vulva lactica, Enteromorpha compressa, and Cladophora fascicularis. Oil extraction was performed in algal biomass. Optimization of extraction parameters was carried out, and high oil yield was obtained by optimizing the moisture content, particle size, stirrer speed, temperature, time, and solvent to solid ratio. After optimization, 10.88% gram per gram oil extraction yield was achieved. Enteromorpha compressa algal oil with high free fatty acids used as a feedstock for biodiesel production. Two-step process was developed. The acid esterification was carried out to reduce FFA content, followed by transesterification. In sum up, biodiesel could be produced from marine macroalgal biomass, and it could be an efficient source for renewable energy production. Now I would like to start my presentation. My title is the role of computational tools in optimizing the production of biodiesel from marine macroalgal biomass. First, we shall see the objective of our production. The production of biodiesel from various sources such as crab seeds and marine macroalgal biomass. Usually the biodiesel will be produced from initially with the crab seeds. Later on, they have tried with that microalgal biomass. At present, most of people are concentrating more on this macroalgal biomass. The present fuel system is from that petroleum based. For example, the compressed natural gas, liquefied petroleum gas, petrol, gasoline, and diesel. They have been produced only from that petroleum crude oil. So this will be called like as a petroleum based fuel system. What are the disadvantages of petroleum based fuel system? That we shall see that. While we are processing this petroleum crude oil, the crude oil will be containing enormous amount of sulfur. While we are processing that time, the sulfur dioxide will be getting produced. By combining this sulfur dioxide along with that rainwater, that will become like an acid rain. the acid rain will be giving enormous human health problems. So we could not use this. And day by day, the price is also increasing like environment, like anything. And also this will be giving environmental hazards. Due to this problem, we need the best alternative fuel, mainly for transportation. First, we shall see what is biodiesel. Biodiesel is that a long chain fatty acid. This will be a long chain fatty acid, and this will methyl ester. So this is the structure of this biodiesel. What are the advantages of biodiesel when compared to petrol diesel? We shall see that. So this can be domestically produced and it can be used in most of the diesel engines. And it is giving less air pollutants, less greenhouse gas emissions. That means CO2. Whatever the CO2 which is produced during the cultivation of algal biomass, the CO2 will be consumed. This is a major advantage. Because of the emission of CO2, the global warming problem is also being there. At present, we are facing this problem. So if we are cultivating more amount of this algal biomass, the CO2 mitigation will be taking place. The next one, this could be like as a biodegradable, non-toxic, and safer to handle. For the purpose only, we are trying to utilize this biodiesel when compared to that petrol diesel. Now we shall see why biodiesel, not the neat oil. That means why can't we use this neat oil as such? But the neat oil will be having more viscosity. If we are going to use this neat oil over the engine parts, small nozzle, that neat oil cannot penetrate. For the purpose only, we could reduce the viscosity. In this case, we must treat this vegetable oil along with the alcohol in presence of various catalysts to convert that into biodiesel. And we can get that byproduct as a glycerol in this particular process. So this vegetable oil, that is alkali esters biodiesel, will be having this viscosity around 4 to 5 mm squared per second. This neat oil will be having 27 to 35 mm squared per second, but once it is converted into biodiesel, the viscosity is reduced as 4 to 5 mm squared per second. So this could be utilized especially over that engine parts, and this could be also easily processed through that small nozzles. After producing the biodiesel, we must check the quality of that oil. So first one, we shall see ASTM method. In the ASTM method, we have to check various property of that oil. And the method also will be present over there. Through this method only, we must check all the property. And correspond to the units, we have to check the values. The values should be within that limits. If the values are within that limits, we can confirm that we can produce a biodiesel. This will be called like as a ASTM method. The next method is the European standard method. So in this method also, we can check various property with the use of various testing procedures. With that units, we have to check the values. The value should be within that limit. If it is within that limit, then we can confirm that we can produce a biodiesel with the use of this European standard. After producing the biodiesel, we must use it in the engine. But in that engine, we'll be getting emissions. The emissions also we must check. The first one is, you can see that graph. The first one is that petrol diesel, and the second one is that biodiesel. The blue color, you can see that this is the NOx emission. In that biodiesel, there is a slightly increase in the emission of NOx, even though that the exhaust we can keep that adsorbent, and whatever the NOx which is emitted from that exhaust, that can be also absorbed and we can remove it. The next one, you can see that particulate matter emission. The particulate matter emission in that biodiesel is found to be very less when compared to that biodiesel. So this is the major advantage. So the biodiesel will be having less particulate matter emission when compared to that petrol diesel. So this is a major advantage of using this biodiesel when compared to that petrol diesel. Feedstock selection. Usually, the various feedstock which are available over there, especially for biodiesel production. But we have to see these various factors. Based upon the factors only, we must select the source. First one, it should be available more. Next one, the productivity also found to be more. The lipid percentage present in that source also should be more, and should be somewhat cheap, and should be utilized for other purpose. Based on this criteria only, we must select the source, especially for biodiesel production. In my laboratory, we have also produced biodiesel using various edible seeds such as mustard seed, castor seed, palm fruit kernel. From this, we have also produced the oil, that the oil will be utilized especially for biodiesel production. Even we have also produced a biodiesel from sunflower seed, Adenanthera pavonana seed, and soybean seed. So previously, whatever we have seen, everything will be like as an edible one. This is a non-edible seed. From the non-edible seed also, we can produce a biodiesel. We can utilize that Cetroba curcus, Pungamia pinnata, Madica indica. So these are all the seeds we have utilized especially for biodiesel production. But these non-edible seeds are already utilized especially for medicinal purpose. So we could not use it especially for the biodiesel production. We could also utilize neem seed and also cotton seed, especially for the biodiesel production. So the above seed oil could not be utilized especially for biodiesel production because they are utilized especially for food purpose and also medicinal purpose. So we cannot be given permission at present to produce a biodiesel from this seed. So at present, most of the people are concentrating more on this micro and macro algal biomass, especially for the biodiesel production. So by laboratory, we have also produced the microalgae, especially for the biodiesel production. We have also utilized nanochlorobacter species and the botrytis species, especially for biodiesel production. And these are all the microalgae can be cultivated in different types of reactors, such as a flat plate reactor, annular reactor, and a tubular reactor. This type of reactors could be utilized especially for cultivation of this microalgal biomass. So different types of the photobioreactors have been also utilized especially for microalgal cultivation. So this will be like as an annular reactor, this will be a flat plate reactor, and a tubular reactor. This type of reactors have been utilized especially for cultivation of microalgae for the biodiesel production. But the main problem of that microalgae is that production of the biomass. Even though we are using enormous different types of the bioreactor, but even though we could not produce more amount of the microalgal biomass, that is a major problem. For the purpose only, we have utilized the marine macroalgal biomass. First time in my laboratory, that world also we can see that we have only produced a biodiesel from the marine macroalgal biomass. We have also utilized Vulva lactica, Enteromorpha compressa, Cladophora flascoresis. This type of marine macroalgal biomass we have utilized especially for biodiesel production. So the macroalgal biomass can be also utilized for other biofuel, such as bioethanol, biohydrogen, biogas can be produced from the algal biomass through fermentation. The biodiesel also can be produced from algal biomass through transesterification. And the biochar, jet fuel, synthesis gas, and the gasoline can be also produced from algal biomass through thermochemical process. Earlier, I mentioned that the biodiesel could be produced from edible crop seed, that will be like as a first generation. The second generation biodiesel was produced from non-edible crop seed. The third generation, we have to compulsorily produce this biodiesel from marine micro and the macro algal biomass. Now we can see in that seashore side, enormous amount of the marine macro algal biomass available over the seashore side. While the degradation is taking place, this microbial biomass combining along with that this bacterial species will consume the dissolved oxygen present over in that water during the degradation of algal biomass. While we are consuming this dissolved oxygen present over that water, the aqueous system will be getting affected. Whatever the aqueous living beings which is available over there, that will be getting affected during the degradation. For the purpose only, frequently we have to remove all the algal biomass present over in that seashore side. So the two way we are giving that advantage. One is we are removing that algal biomass from the seashore side. Other side, the removed algal biomass could be utilized especially for biofuel production. So these are all being like as an advantages over in this using of algal biomass, especially for the biofuel production. Sequence strategy for biodiesel production. There are two methods. Other one is two-step process. Other one is a in situ transfiguration process. The two-step process, initially we have to extract the oil from algal biomass. Afterwards, we must go for acid esterification and the base transfiguration process. This will be called like a two process. Most of the people are utilizing only two-step process, especially for biodiesel production. Next one is the in situ transfiguration process. In this process, simultaneously, we have to extract the oil and the transfiguration also will be taking place in the same reactor itself. So this will be called like as an in situ transfiguration process. But here, most of the people are not utilizing because there is a loss of this oil. For purposes, most of the people will not be utilizing this in situ transfiguration process. Elements of the research. First, we have to collect that algal species from that seashore side. Afterwards, we have to process this algal biomass. We have to extract the oil. through two-step process. Then afterwards, we could convert that into biodiesel. Objective of our work. First of all, we hope to extract the oil from marine macroalgal biomass. Afterwards, we hope to check the free fatty acid content. The free fatty acid content is more, we hope to reduce the FFA, that means free fatty acid. For the purpose only, we must do this esterification process. Next, we hope to convert that esterified oil into transesterification process. Through the transesterification process, again, we could convert that into biodiesel. Once we are getting the biodiesel, that must be characterized with the spectrometry, chromatography, and thermal analysis. And finally, we have utilized this biodiesel especially for other purpose. Materials and methods. We have also collected the Enteromorpha compressa, Vulva lactica, Clarapa peltata from that Rameshwara Mandapam South Coast area. After collecting that algal biomass, we have washed with normal water. Then this is shade dried and oven dried with 40 to 45 degrees centigrade. Afterwards, when pulverized, the required size of that particle, size of this particular algal biomass was utilized especially for biodiesel production. Pre-treatment of algal biomass. This is very, very essential because we have to break the cell wall. Once we are breaking the cell wall, easily we can extract the oil. After extracting that oil, this could be converted into biodiesel. For purpose only, various pre-treatment methods were utilized, like as an autoclave method, ultrasonication, lyophilization, microwave, bead beater, deep freezing. These are all the pre-treatment methods were utilized especially for breaking the cell wall. Process design for two-step process. Initially, we hope to collect that algal biomass. Then we hope to pre-treat that algal biomass. After pre-treating that, we hope to extract the oil. During the extraction itself, we hope to optimize various parameters. The optimization of extraction parameters must be carried out, especially for increasing that oil yield. After optimization, we hope to purify that algal oil. After purification, we must do that esterification. So the esterification itself, we hope to optimize various parameters. Esterification kinetics also we must do that because we hope to design the reactor. For the purpose only, we must find out the esterification, that K value. Then afterwards, we must do the transfiguration. During the transfiguration itself, we hope to optimize various parameters. The transfiguration, during that time, we hope to do the kinetic study. After doing that kinetic study, we must purify that biodiesel. After purifying this biodiesel, we must also characterize this. This is the process designed for two-step process. So algal oil extraction. This experimental setup, we can see that 3-neck round bottom flask must take that pulverized this algal biomass along with that solvent, and also with the magnetic stirrer we must heat that. For heating, the solvent will be contacting along with that algal biomass, then solvent will dissolve that oil present over that algal biomass. When rising the temperature, the solvent will be coming like as a vapor on the top side. During that condensation, this solvent will be again recirculated toward this particular round bottom flask. In this way, continuously, the oil will be extracted from that algal biomass. So for maintaining the temperature, we must insert this thermometer. Through the thermometer, we can sense what is the temperature which is being there inside. And this is sample port. To the sample port, we can collect that solvent, how much amount of oil is extracted, that also we can collect. And also in that particular experiment, we have also utilized a combination of various solvent and solvent mixture, especially for increasing this oil extraction yield. So this is the oil extraction stock setup. As the previous one will be like as a batch process, and this will be like as a continuous one. Here you can see we are using that single neck round bottom flask. Above that only we are keeping that thimble. The thimble inside we are keeping this condenser. So in that round bottom flask, we are taking the solvent and raising the temperature. We are raising the temperature, the solvent will be coming like as a vapor on the top side. That will be getting condensed. Once the solvent is getting condensed, that will be allowed to contact with that algal biomass. Then the algal biomass will be in a powder form. The oil will be easily extracted by that solvent. The solvent plus oil will be tried to come through this siphon tap. Then it will be getting collected at that bottom side. Again, we will rise the temperature. The solvent alone will be coming like as a vapor. Then that will be getting condensed. That will be allowed to contact with that algal biomass. So to overflow, this will try to come at the bottom side. So oil will be continuously collected on the bottom side, extracted oil, and this solvent will be coming like a shaker that will be alone will be circulated. So in this way, only continuously we are extracting the oil from algal biomass that will be collected at the bottom side. After some 8 hours, we can dismantle it, and by raising the temperature, the solvent will be coming like a vapor on the top side, and we can collect it separately. Only that the oil will be there at the bottom side. That oil could be utilized especially for biodiesel production. This will be a single neck, and this will be a double neck. The double neck, we can provide it to set of thimbles. Next one, the algal oil can be also utilized for a food purpose. So now we can see that in the fermenter or the reactor, we can utilize whatever the waste which is generated over the agro-food industry, and the strain can be cultivated over this fermenter. Then after getting the biomass can be collected separately, and the oil could be extracted from that algal biomass, and in that oil itself will be having pigments, and the pigments also we can remove that. The remaining algal oil can be utilized for medicinal purpose. The next one, the residual biomass will be available over there, that will be utilized for other purpose. Then the required strain will be inoculated, incubated in the fermenter, and the biomass could be separated through the centrifugation. Oil could be extracted. The extracted oil can be utilized as saponification and the esterification. Finally, that oil could be like also taken for the vitrification. The vitrified oil will be having aromas of omega-3 fatty acid, and that could be utilized especially for medicinal and food purpose. The lipid extraction. So first we must cultivate that algal biomass. Afterwards, the cell must be disrupted. Then lipid must be extracted through vitrification, that could be utilized especially for other purpose. So in summary, we can see that the PFAR, whatever that oil we are getting, that will be considered as a PFAR, that play an important role in the human health related to circulatory system, immune system, and also nervous system. So whatever that algal biomass which is available over there, that algal biomass can be utilized especially for extraction of oil. That oil could be also utilized especially for this food purpose. So whatever that omega-3 fatty acid that we are getting, that will be having enormous advantages that have been made like as a list. Now we can see that this chetroba oil and this algal oil both can be mixed, that will be called like as a bio oil. The bio oil will be taken over the transferification reaction. The transferification reaction, we can see that this 3-neck reactor. Then here we are having that agitator. Thoroughly we have to mix that at about 6 degrees centigrade. The oil could be converted into biodiesel in presence of this methanol and also the catalyst. Once we are getting the biodiesel can be utilized especially for running the engine. So this experimental setup, we can see that this is the ultrasonication reactor. Inside that we are having the water bath. The water bath only we are going to keep this 3-neck round bottom flask. Here at the center, you can see that there will be an agitator, and at the second neck we are providing this condenser, and this will be like as a sample port. There itself we can see that what is the temperature with the use of this ultrasonication reactor. Now we hope to see that what are things to be added over inside of this ultrasonication reactor. We shall see that. The acid-base catalyst that could be mixed along with the methanol. This will be called like as acidified alkalified methanol. So this alkalified acidified methanol will be added along with the algal oil. Everything will be taken inside this round bottom flask. After keeping it over inside the round bottom flask, this will be again kept over inside of this ultrasonication reactor. The use of this, we can continuously monitor the temperature and also the sonication. With the use of the stirrer, thoroughly we can mix that, and with the use of this condenser, whatever that this methanol will be coming like as a vapor can be condensed and again also recirculated. Once that biodiesel is formed over there, this could be taken over in that this funnel. That funnel, we can see that the bottom side will be having like as a glycerol, top side will be having like as a biodiesel. After 8 hours, if we are keeping that, we'll be getting 2 layers. Bottom layer will be a glycerol, top layer will be a biodiesel. By opening that valve at the bottom side, the glycerol will try to come through that gravity separation. So biodiesel will be there in that separating funnel itself, that we can use it for biodiesel. So this is the normal setup. So this is a round bottom flask. Inside that only we are keeping that oil along with that methanol and also the catalyst. Right? By raising the temperature, the biodiesel will be formed. The methanol will be coming like as a vapor. For the purpose only we are providing this a condenser. The use of this peristatic pump, we can circulate the cold water or that annular side. So in this way, we can maintain the setup, especially for biodiesel production. Once the biodiesel is produced, this could be transferred into that separating funnel. The separating funnel, you can see that bottom side will be having that glycerol, top side we are having the biodiesel. By opening this wall to the gravity separation, this glycerol will be allowed to collect on the bottom side, and top side we are having that biodiesel that will be transferred and utilized for other purpose. During the extraction, we hope to optimize various parameters such as moisture content, particle size, mixing intensity, extraction temperature, extraction time, solvent to solid ratio. These are the parameters could be optimized, especially for increasing that oil extraction yield. Once we are getting that algal oil, we must also characterize that. For the purpose only, we are using this GCMS analysis for free fatty acid composition. And the functional group present over that oil also could be analyzed with the use of this HNMR analysis. Physical property and the chemical property also we could analyze. So this is the reaction which is being there, especially over in this acid acidification and base transfication process. So this free fatty acid will be converted into that methyl esters in presence of methanol and acid as a catalyst. Okay, the byproduct will be the water. And you can see that in the transfiguration process, the diglyceride will be treated along with the methanol in presence of base as a catalyst. We are getting that biodiesel, and the byproduct will be the glycerol. This is the reaction which is being there, especially in the case of esterification transfiguration process. During the esterification process, we hope to optimize various parameters such as catalyst concentration, methanol to oil molar ratio, mixing intensity, reaction temperature, reaction time. So these are the parameters should be optimized, especially to get the maximum oil conversion into biodiesel. Optimization study, especially for transfiguration process. In the transfiguration process also, we have to optimize various parameters to increase that conversion. We have optimized the parameters such as catalyst concentration, methanol to oil molar ratio, mixing intensity, reaction temperature, and reaction time. Once we are getting the biodiesel, we have to also evaluate that. For the purpose only, we have to use various instrumental analysis such as spectroscopy, that is a near infrared spectroscopy, then high performance thermal chromatography, and thermogrammetric TGA and DA analysis. This type of analysis could be utilized especially for a biodiesel. And physicochemical characterization also we must do that. For example, cetane number, density, viscosity, water and sediment, clouded pour point, flash point, copper strip corrosion test, sulfur ash content, distillation rate, sulfur content, free glycerin, total glycerin, carbon residue, acid value, and phosphorus content. All these things that we could establish after getting the biodiesel. Kinetic study is also very, very important. The first order kinetics, we go to follow this. The second order kinetics, we go to follow this expression, especially for finding out the K value. Through the K value only, we go to design the reactor. For purpose only, we must follow this kinetic study. Result discussion. Previously, we have established all the experiment. Now we go to see whatever the result we have obtained. Already I have told you that we are utilizing the pre-treatment method, especially for breaking the cell wall. We have utilized various pre-treatment method. This pre-treatment method, this ultrasonication has given that maximum oil extraction yield. And within that sonication time of 5 minutes, we have obtained maximum algal oil yield. Effect of various solvent system on biodiesel production, especially extraction. In this case, diethyl ether and that methylene chloride in hexane has given that maximum oil extraction yield of 7.33%. Optimization of various parameters on algal oil extraction. In this case, the moisture current of 5% has given that maximum oil extraction yield, and the particle size of 0.149 has given that maximum oil extraction yield. And also you can see 600 RPM, 55 degrees centigrade within 160 minutes, and also the solvent to solid ratio of 8:1 has given that maximum algal oil extraction. Once we are getting algal oil, we have also characterized that the characterization we can see that the polymetic free fatty acid content was found to be more relatively around 41.63%. And also we have determined the average molecular weight that is corresponding to 824.50 gram per mole. This is very, very important. Based on this parameter only, we can add how much amount of oil with that methanol. So these type of calculations, this average molecular weight is very, very essential. Acid value is also very essential. The free fatty acid content can be obtained from that acid value only. Usually the free fatty acid content should be less than 3%. If it is more than 3%, then we go for this esterification. If it is less than 3%, directly we can go for this transterification process. Then we have to analyze this algal oil for HNMR studies. In this case, the 4.2 corresponding to that area under this curve also we have to see that. So this is more important. This 4.2 corresponding to whatever the peak which is available over there, we have to calculate how much amount of this algal oil is present over there, especially for the biodiesel production. Now we can see that corresponds to 4.142 only. We are having that alpha methyl group of glyceride. This is the main thing that we have to concentrate. This only will be converted into biodiesel. So correspond to 4.2, we have to find out whatever the area under this curve. This is coming around some the ppm level corresponding to 4.142. The next we go to optimize various parameters, especially for esterification. In this case, we can see that the influence of the catalyst concentration around some 1.58% has given that maximum oil yield, and the methanol to oil molar ratio of 12:1 has given that maximum algal oil. And this 400 rpm, 60 degrees centigrade, and 20 minutes of duration has given that maximum acid esterification yield. Kinetic study also we have carried out. In the case of esterification process, we can see that the R squared value was found to be closer to 1, around some 0.992 in that first order kinetics, when compared that second order kinetics of R squared value 0.980. From this, we can conclude that this esterification parameters found to be fitted very well with that first order kinetics when compared that second order kinetics. Next one, transformation process. In transformation process also, we have optimized various parameters. First one is the selection of catalyst. The case NaOH catalyst has given that maximum this is transformation oil. Then the catalyst concentration of around some 1% has given that maximum oil conversion through transformation. The methanol to oil molar ratio of 9:1 has given that the maximum conversion through transformation. And 600 rpm at 60 degrees centigrade within the 12 minutes, the maximum conversion was obtained the use of this transformation process. Kinetic study also we have established. In this case, we can see that this is the first order kinetics and this is the second order kinetics. In this case, the R squared value of the second order kinetics was found to be closer to 1 when compared to that first order kinetics. This shows that this transformation parameters were found to be fitted very well with the second order kinetics when compared to the first order kinetics. So we can say that this kinetic study found to be fitted very well with that this transformation parameters with the second order kinetics. Characterization of enteromorpha biodiesel. Once you get the biodiesel, we have analyzed through this HNMR spectroscopy. In this case, you can see that this is for that biodiesel and this is for that this oil. This is biodiesel and this is that oil, extracted oil. Here you can see there is a peak. This is a triglyceride. The disappearance of this triglyceride shows the biodiesel production. Right? The use of this analysis, we can confirm that we have produced the biodiesel. And this is the chromatography. The use of this HPTLC, we can see that this is for that oil and this is for that biodiesel. This is a monoglyceride, diglyceride, a triglyceride. And that fourth peak which is be there, but this fourth peak is not available over there. But for this biodiesel, we can see that the HPTLC analysis, this peak height has been reduced. The monoglyceride peak has been reduced. Diglyceride peak also is reduced. Then the third one, the triglyceride peak is also reduced. Apart from that, one more peak which has been formed over there. This is nothing but the biodiesel. Based upon this HPTLC analysis, we can confirm that we have produced the biodiesel. This is the TGA analysis. We can take that oil and we go to rise the temperature. The x-axis we can see there is an increase in the temperature and there is a reduction in that weight. At about 30 degrees centigrade, we can see 100% weight is there. For rising the temperature, the evaporation of the oil is taking place. That time the weight has been reduced. But even the temperature range of 161 to 289 degrees centigrade, The reduction in that weight of the oil will be confirming the biodiesel formation. The use of this TG analysis, we can confirm we can produce a biodiesel. And physical-chemical characterization. With the use of this ASTM method, we have analyzed various properties such as a flash point, kinematic viscosity, water sediment density, cetane number, cloud point, acid value. All these properties were checked with that various ASTM method. And also we have obtained the values along with the units. And we have also checked all the values are within that ASTM limits. From that we can confirm we have produced a biodiesel. This HNMR spectroscopy after producing the biodiesel. Corresponding to 4.2, we are having the triglyceride. Now we can see that at about correspond to 2.3, we are getting that FIB. That is nothing but the biodiesel. Once the biodiesel is formed, the peak under this will give that formation of a biodiesel. Under 4.2, there is no any peak at all. So whatever the triglyceride which is present over there, everything will be converted into that biodiesel. While you are seeing that oil, this corresponds to 2.3, there is no any peak at all. But after converting the biodiesel, after 2.3, we are having that peak. From this peak under this curve, we can say that how much amount of the biodiesel is formed. Finally, we can conclude that the algal oil extraction yield was calculated around some 11.14%. The conversion was found to be 98.76% from the enteromorphic compressor marine macroalgal biomass. And the esterification reaction followed the first order kinetics, and the transfication followed the second order kinetics mechanisms. The fuel properties were found to be satisfied, and they were found to be within the limits of ASTM. Now this we can confirm that the marine macroalgal biomass was utilized like as a raw material for the biodiesel production. So thank you slide. In this case, you can see that we can cultivate the marine micro and the macroalgal biomass in presence of sunlight, whatever the CO2 which is coming like as an emission from this engine, and also in the presence of sunlight and water, we can cultivate this algal biomass. Once the algal biomass is produced, that could be taken for extraction. After extracting the oil, the remaining biomass can be utilized for other purpose. Now that extracted oil could be converted especially for the biodiesel. During that biodiesel production, the glycerol will be coming like as a byproduct. This could be utilized especially for medicinal purpose and also this cosmetic purpose. The produced biodiesel can be utilized especially for running our engine. During running that engine, enormous amount of CO2 will be getting produced. That CO2 could be utilized especially for cultivation of algal biomass in presence of sunlight and water. Right? The mitigation of CO2 is also very, very essential. Already I have mentioned that if for producing more amount of CO2, the glowing problem also will be coming. For the purpose only we have to mitigate this CO2. So the CO2 could be utilized especially for cultivation of this algal biomass. Once the algal biomass cultivated, from that we can extract the oil. That oil could be utilized for biodiesel production. Again, the biodiesel production can be utilized for running our. This will be like as a cyclic process. So the green energy, one side we can produce, at the same time we can reduce that CO2 content. And also whatever that remaining biomass which is present over there, that will be containing enormous amount of protein that can be placed for other purpose. When glycerol also will be produced. At present in my laboratory, the people are utilizing this glycerol especially for the production of methanol. That methanol can be utilized like as a one more raw material especially for the production of this biodiesel. So whatever the waste which is generated, everything can be utilized especially for biodiesel production. So through this we can produce like a green energy, and also we can save this our. Thank you very much for giving me this opportunity. For having any doubt, you can also discuss along. Yes, please.
Thank you, sir.
This is
Thank
open
you
forum.
for,
Yes, sir.
Thank you for your informative session. You have
Yes,
provided a lot of information about production, purification, and characterization, and application of
yes, sir.
your diesel from the algal biomass. A very informative session, sir. Thank you for your presentation. So now,
Actually, in the computational tool, yes, sir. The computational tool we can use for optimization of various parameters. We can use various software. RSM, we are having that. With the use of this RSM, we can optimize various parameters easily. And also, instead of that doing that process elimination method, the use of this RSM, right? Simultaneously, we can vary all the parameters and you can optimize various parameters. That is a major advantage of using this RSM method. So computational tools also we can utilize, especially for that our extraction and also the biodiesel production. In my laboratory, one student has also established a research work through that computational tools. But we could not show it everything over here. For purpose, I am concentrating more on this, especially the normal extraction and the biodiesel production procedure. Okay, sir. For having any doubt, you can also discuss along with me, please.
Yes, sir. Participant, if you have any doubt, you can ask to sir or put in the chat box. This is the time for you.
Any doubt is there, please?
So I have one doubt, sir. You have
Yes, sir.
tell about the reactor for algal production. So any possibility for open system for algal production for biodiesel extraction?
Yes, sir. Actually, one side already have mentioned that mitigation of CO2, right? So if you are allowing that algal biomass to cultivate, even that room itself, sometime at present, people are keeping that algal biomass, they are cultivating that. Whatever the CO2 which is available over there, the CO2 will be absorbed by that this algal biomass. That is a major advantage, and the CO2 also will be getting reduced. One side, the open pond itself, we can cultivate this algal biomass. In the room itself also we can cultivate, right? That is one side. Other side, you are asking one more question, extraction. The extraction can be also done the use of various methods. Already you mentioned that the oxalate apparatus also we can utilize that, especially for extraction of algal oil in a continuous operation. But the batch operation also we can extract the oil, right, sir? Any other doubts, sir, apart from that?
Thank you, sir. Thank you for clear my doubt. So one
Yes.
question in the chat box from Dr. Bhuvaneshwari ma'am. How efficient biodiesel production from algae cost effective or not?
Actually, when compared to seed, it's very difficult from that algal biomass. From that seed, easily we can extract the oil, but extraction of oil from that algal biomass will be somewhat difficult because it's a technology. Everybody cannot utilize that one. So we have to do it in a proper way to extract the oil, right, sir? So it is a separated technology which is being there. So that also we have to follow that. While we are just extracting the oil, charring effect also will be taking place. That also we have to avoid it. With the less temperature, only this reaction must be also carried out, especially for extraction of oil. Once you're extracting oil, easily that we can convert that into biodiesel. So during the extraction, we must take too much care. We should not rise too much of temperature because a charring effect also will be taking place sometime. Right, sir? So through that permutation competition only, we must do that experiment for the extraction of oil from that algal biomass. Yes. And the cost will be also somewhat in that higher level when compared to that normal procedure. But at present, you can see that we are having the crude oil. Easily we are producing this the petroleum crude oil. That is a petroleum from the petroleum crude oil. Easily we are producing this diesel. But after a period of time, this crude oil may not be available over there. So that time compulsorily we go to produce a biodiesel. Then that time everybody will be concentrating more on this algal biomass, especially for production of biodiesel. That time the cost also at present biodiesel people are producing from this seed only. The use of this crop seed, they are selling it at the market cost around some 60 to 75 rupees at present. Even that the Gujarat side, people are selling that in that black market. But the production of biodiesel with the use of algal biomass would be somewhat too difficult at present. After a period of time, if you are unable to get crude oil, that time most will be concentrating more of this algal biomass only for the production of biodiesel. Any doubt is there apart from that,
Thank
sir?
you, sir. Yes, the participant, if you have any doubt, you can ask to sir or put in the chat box.
So apart from that biodiesel, at present people are utilizing this algal biomass for various purposes. Even that mechanical people, they are utilizing this algal biomass instead of this thermocol, because for avoiding this radiation loss, they are using this particular biomass instead of like as a thermocol. Thermocol may not be like as a biodegradable, right? At present, the people are using this type of algal biomass like as an instead of this thermocol. This type of research is also going on. One person was discussing about that. So this algal biomass can be utilized for various purpose, medicinal purpose, food purpose, that insulation purpose, like that. Various application at present people are using this algal biomass. Even you can also try for the different purpose. And the algal biomass can be also easily cultivated. It is available like as a plenty over the seashore side, right? That could be utilized for other purpose. When you are cultivating this algal biomass, the CO2 production also will be getting reduced. So for this purpose only, we are encouraging all the people to cultivate this algal biomass. So any doubts apart from that?
Yes, sir. So
Yes.
participant, we will going to wind up the session. So thank you, sir. Thank you very much for your passion. Thank you very much for your wonderful and informative presentation regarding biodiesel production. So thank you for your session, sir. Thank you.
Yeah. Thank you very much for giving this opportunity, sir. Whenever you're having any doubt, you can also discuss along with him, especially for this algal biomass utilization for various purpose.
Sure, sir. Sure.
Yes. Thank you, sir.
Thank you very much.
Thank you.
Participant session 8 feedback link provided in the chat box. So kindly all of you fill in the form for your attendance and feedback about this session. Participant, kindly fill the feedback link. It will provide in the chat box. Thank you.